So what I'm going to tell you about today is what we do. I head up the Structural Genomic Consortium, which is a public-private partnership, as she said, which is funded by 8 pharmaceutical companies, Genome Canada, Ontario, the Wellcome Trust in the United Kingdom. We have two laboratories, one at the University of Toronto, one at the University of Oxford. There's about 100 scientists in each one. We collaborate with lots and lots and lots of people. And our mission is to generate and put into the public domain, with never filing for a patent, chemical inhibitors, recombinant antibodies, and 3-dimensional protein structures of human proteins of therapeutic interest. Each company donates us $8 million to put stuff into the public domain and never file for a patent. Okay, it's a new world, and it's a fun world. So, why are we doing this? Well, I'd like to sort of just frame it for why can't we invent drugs? Right? More and more money is going into drug discovery, and the number of new medicines is not coming out. Currently, pharmaceutical companies spend $100 billion a year in research and approve 20 new medicines. If you do the mathematics, that's $5 billion per new medicine, and that's not even a really novel one. There's probably about 5 to 8 novel ones every year. Okay? And since that number keeps going down, pharmaceutical companies say research is a waste of money, and therefore they close sites. As you guys have seen in Quebec, in the United States, since 2009, 20 pharmaceutical sites have closed. In the EU, 16 since 2009. It's a worldwide phenomenon. It's nothing to do with Quebec. The pharmaceutical company says investing in research is a waste of money. But why? Why can't we invent the medicines? We spend literally hundreds of billions of dollars. Look at you, you're very, very smart. We discover things every day. What's the disconnect? Why can't we do this? Well, it turns out all of our beautiful ideas in test tubes, when we finally test them in a human being, new ideas, 19 out of 20 don't work. And to get to that point, so-called proof of concept, it costs about $50 million in 7 years. Okay? So that is the terrible part of drug discovery. It's not that we can't make molecules, it's we don't know what to make them to. And when we have a great idea and it works in our test tubes, it doesn't work in man. But why? We seem to know a lot. What's the problem? On this axis here are the 518 kinases in the human genome. These are drug targets. They're fantastic signaling molecules. Everyone has a name. So you can go into PubMed from 1950 to the present and ask how many papers are there on each kinase, where the kinase is mentioned in the title and the abstract or the keywords. And that's the black bars. So you can see that we get billions, 100 billion every year, and most of it is focused on 10% of the kinome. The genome, apart from all the other things it did, it really let us quantify how much work we do on certain things. You can't do that in astronomy, the stars are infinite. You can't do that in chemistry, there's 10 to the 80 molecules to make. With the genome, it's there, and we can do this kind of analysis, and it's kind of scary. Now you might say, professors will try to argue, we're so smart that we knew the important ones. But then you do RNAi studies, or you do deep sequencing of cancer, and you find out that the kinases that are very important for interesting biology don't overlap with the ones we're working on. These are largely historical. And it's nobody's fault, remember. It's perfectly explicable, because if you write a grant, as the students will in about 4 years, you'll be writing them, the first 3 pages are background and significance. How in the world do you write background and significance for kinase 401 where there are no papers? How are you going to explain to the reviewers that you should get money to work on it when you can't explain why? Whereas if you work on kinase 20, you can tell a great story. Right? So-and-so knocked it down, the mouse did this, and the mouse and the cells did this, and I predict this, and you draw balls and circles, and this upregulates that, and you'll get your grant. You'll get your grant because everyone around the table works on that protein. And, well, that's a very important protein. We should fund that one, right? And if you work on this, you'll hear the word fishing expedition. It's not elegant. It's not elegant to work down here. It's really elegant to tell that story, right? And we get funded on stories. You hear people, we need to write a good story in the paper. It's a story, right? It's sort of strange, right? We should be in literature. The other interesting thing is that why do we go into science? And it's a human nature to be accepted by peers. You want to get invited to go give a talk in Barcelona? Don't work on kinase 401. No one on the organizing committee knows about it. No one cares about it. They all work on kinase 20. You want to get invited to Spain? Work on kinase 20. You're there. It's a serious problem, and unfortunately it's not getting better. Right? You see, you work on the same protein, and there's always something interesting about it, and you work on it, and your students work on it. They get their own labs, and they work on it. And you have a little community, then you have a meeting on that protein, and it's all exciting. I'll go to Crete, and you get together on that protein. And no one's working out here, right? Now you put a molecule into man, and you come to me, and I say, trust me, I know how human body works, take it. Because we've gotten convinced, the Harvard professors with the low bow ties and the lab coats have told you they know a lot, of course, about this tiny, tiny, tiny little area. But the real proof of understanding is when you try and intervene in a human and it doesn't work. So they can't be right no matter how smart they sound. And it does affect industry as well. So look at this. This is patenting from the last 3 years. Take a step back, don't look at the details. Doesn't the pattern look sort of the same? And remember these, a lot of these were 1990 specials. And we're still publishing on them, so they're still working on them. And the same thing, industry is, they're justifiably data-driven organizations. Justifiably, they're going to invest $10 million in a drug discovery program. You better be sure you want to know the target and the disease indication, etc. Where do they get that? The academic literature. Who do they talk to? The key opinion leaders, the Harvard guys with the bow ties, right? Everyone talks to the same Harvard guy with the bow tie, though, and you start working on the same stuff. And as a result, where do we as society is the biggest bang for the buck? Where is the innovation? Where are we going to get cures for Alzheimer's and schizophrenia and all the diseases that we really want cured? Maybe not here, but more likely, there's going to be some gold out here, and none of us can work on it. And it's distressing. And so this was the driver for the SGC. What we started to do is, how can we get to the unknown? But first, you got to know how we're going to do it. So the problem is that neither industry nor academia, it's nobody's fault, remember, but the system we put in place doesn't allow us to go into the unknown. And that's in part the way in which we reward scientists and get our recognition. We get our recognition by our friends telling us what a good job we're doing. Well, if we're working alone, they're not going to tell us that. You have to work where other people are working, gain your credibility, be a little super stud in that little area, and you go home feeling good. You're not helping anybody, but you feel good because all your friends tell you you're good. And industry does rely on us to be innovative. So can we do anything about it? This is the important thing. If the molecule is there, we will do the experiment. There's a protein down there mutated in mature onset diabetes of the young. There's circadian rhythm proteins, Parkinson's proteins. We don't work on those. We work on the ones that we can do the experiment. It's pretty logical. Your student has 3 years to graduate. You've got to give them a safe experiment. Making one of these molecules probably costs $5 million in 3 years. You just don't have the resources. It's much easier. And again, we tell stories. If you have an inhibitor of mGluR5, you're going to do an experiment on mGluR5. I think mGluR5 is involved in blah, 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 fill in the blank. Right? You can make that hypothesis because it's pretty easy. That's what we get really good at, right? A geneticist connecting dots. You can find any way to connect two things. But the bottom line is, I think that research tools, the ability to do research, actually have a more dominant effect than biological interest. But evidence shows that you've got to give them away with no lawyers involved, and only then will professors use them. And I think this is an interesting way now to manipulate the world. When you hear governments trying to tell professors you should work in this area, boy, do we get angry, right? Nobody tells me what to do. I am too smart for you, Mr. Government. You need to give me money and let me go and be free. Now, of course, when that happens, what do? We go to the same area and work on the same thing. So I figure if we make tool compounds for these, We don't have to convince professors to work there. It's like flies on you-know-what, right? You throw out the compound, and they'll all start working on them. And so, this is, I thought, a good strategy, but where in the world are we going to get the chemistry? Because as much as there are fantastic chemists in academia, they cannot match the infrastructure and the background and the knowledge that lies in the pharmaceutical industry, where you have teams of hundreds of chemists and outsourcing material and all the equipment you want in the world. It's a machine. So how do you access that machine? So, Tim and I got together and we managed to convince Patrick Valance, who's the head of R&D at Glaxo, that why don't we pilot an idea where we at the SGC, so we make lots of proteins and we do protein assays and we can solve structures of proteins with small molecules. We'll do all that stuff. We'll make cell-based assays, as you heard before, with good Z primes. And then we'll have industry do the chemistry. And when we make a molecule that is a nice inhibitor that works in cells, not that works in animals, because we thought that was too much money for the value, we'll give that compound to the public domain, and anyone can use it. And of course, we get a lot of science done, but how does industry benefit? Industry benefits by all you smart people doing experiments they're not paying for, and they get to watch you publish, and when you link that protein target to the disease, they can decide, you know what, I'm going to start a proprietary, it's a secret program inside my company now, because they did all the work for me. I know that if I inhibit that protein, I'll help in that disease. And that's really what I want to know in industry. The chemistry is not really valuable, because I can always make another chemical. I want to know which target do I want to work on. So right now, we do the biochemical assays, the chemical screening, the structure, the computational chemistry, the cell-based assays, and the chemistry is done in our academic chemistry partners and all these companies. And every single one of them commits to make novel molecule and put one in the public domain without restriction. For the chemists out there, the companies get to keep the SAR. I don't care about that. So long as they put that molecule, a valuable molecule, in the public domain, that's a good deal for the public sector. Once we make the molecules, out they go into the world. And he's in Steph's lab, and they collaborated with a doctor at the Dana-Farber, whose name is Jay Bradner, and Jay was caring for some of these midnight carcinoma patients. And what we did together, and I'll tell you how, was discovered a molecule called JQ1, which turns out to be a very potent inhibitor of BRD4. And when we looked at the cancer, which we put into a xenograft, it knocks the cancer dead. We gave the molecule away to hundreds of laboratories right away. Here you go, no MTAs, just send it away. And other people used that molecule to link the target to two cancers which are much more abundant than midline carcinoma, multiple myeloma, and amylogenous leukemia. That got pharma interested, exactly as planned, right? Let the literature, GSK turned 180 degrees, took the molecule, the program that was going in inflammation and they didn't know what to do, and they get to first in man. For anyone in drug discovery, from the first screen to first in man in 3 years is a phenomenally short time line. If you compare it with Gleevec, which is a very similar case, another fusion protein, a kinase inhibitor that hits the fusion, that took 6 years, and the open sharing did it in 3. Now, of course, not all of this is due to peace and love and sharing and stuff, but about, I think, 2 years of this were interinstitutional negotiations. One doctor actually had to leave the university to another university because they wouldn't allow him to do the deal with this company. And all that time, people are dying while they're doing this little shuffle, right? And so I think that there's incredible scientific value in sharing, and ultimately it will help everybody, scientific knowledge and the patients and the industry, because they'll be profitable faster. But, okay, so that sounds pretty exciting, doesn't it? I mean, we're making molecules, companies are sharing, we're singing Kumbaya, and we're doing all stuff. But there is a slight problem that this competitive nature of us causes. So drug discovery is, in fact, unpredictable and expensive. We go into man, we still don't know what's going to work. It starts with target. Let's work on this target, high throughput screening, selecting a candidate, doing your talks and your phase 1 studies, and then when you finally get to your proof of concept, does it work in the patients with the disease? Oops, no, and it costs a lot of money. That's pretty bad. Imagine all the scientists who work in industry, and you can talk to them. Ask how many have actually worked on a drug, and you'll see them. You go into industry wanting to make a difference, and you ask how many have actually worked that made a medicine, and hardly any. And it's a shame because they're really smart people, and they keep working on this. But what's really, really distressing is that the same protein is being worked on in every company. The same Harvard guy goes around, they read the same papers, they all have the same ideas, and they all work on the same thing. So that's a horrendous waste of money, but even worse, think about it. Let's imagine it's a breast cancer program. When you get on INDs, when you get on new medicines for breast cancer, you have failed the current regimen, right? You're in your last 3 months' life, it's almost a give-up scenario, so you get put on the new drug, okay? The ladies who are on it are taking a risk, of course, but they think, hope against hope, it works. But also, if it doesn't, I've helped. But in fact, that's not quite true. Because if Merck's the first one that fails, Merck's not too keen on telling everyone why it failed. They may say it failed, they may give some hints it failed, but not enough data to stop Astra and Pfizer and GSK. And by the time the second one doesn't work, now we know it's not the chemical, it's the target. We're having ladies getting poisoned to death, all down here. And it's no one's fault. This is the system we put in place to do drug discovery. It's an idiotic system. If you were to invent it now, you'd never come up with this, but this is what's happening. So you have, the problem in drug discovery isn't the smartness of people, isn't it our ideas, it's the systems that make us do stupid things. So why do we have to do this is the question. See, I'm lucky enough now. I've got a lot of the pharma heads of R&D in my room, and we can talk about things. Maybe we can do things differently. Why? I ask the question, why should 20 companies and universities, because universities are getting into drug discovery too, and let me tell you, we're working on the same targets that pharma's working on. Why should we all spend our money on the same ideas when we know 95 out of 100 are going to fail? Right? It sounds cool and competitive, but it's dumb. What if we took the same amount of money and went to this side of the curve and spent it on 20 novel targets, one each? Think about the amount of knowledge that we gain as a society. How many more validated targets would we bring to industry? They'd be more profitable, patients would be saved, and we'd learn more science. This makes sense. So we're coming up with an idea, and it's going to work. We're going to form, and it's a dumbass name, and I'm not even going to tell you what it is, but anyway, we're going to form a public-private partnership to test new targets that we absolutely know no one's working on, and we're going to take it from target to proof of concept in the open, sharing all the data without filing for IP so anyone can use it. And if we do that, the knowledge will be amplified, patient safety will be saved, scientists can work on the problem, and industry is keen. I've spoken to very many senior people. They do it, but they want someone to lead. And the United States won't. The NIH is they've got these rules. If you belong to the NIH, you can't even hold shares in a company. They're so nervous about lawyers and stuff. Europe can't do it. You take one step in Europe, you got to get the Latvian minister and the Lithuanian minister and the Portuguese minister all to sign up to the agreement. You can't do it. Canada can though. And so, I think we are, through this model, really thinking about new ways to help the world discover medicines, and even pharma, which is a really IP, a patent-driven ecosystem, or whatever. They're buying into it. And I think if we can push this to a proof of concept, I think it can work as well as benefit other sectors of the economy. But we need Canada to lead, in my opinion. If we spend $1, are we going to spend it being the 100th dollar on the same problem, competing with the Americans and the EU? We may feel better, we're competitive, right? And we may beat the person by 1 month, right? Or we could be the first people to work in a whole new area. Because in reality, right now, if we keep going, if everyone in Canada got hit by a bus tomorrow, it would not affect world science one bit. Every experiment we're doing is being done elsewhere. We all look at the same literature. There's a lot of smart people out there. Guaranteed, every one of us is in a race with somebody because we're doing the exact same experiment. Now, we win sometimes, we lose sometimes, But in reality, if all of us disappeared, the same stuff would happen. So, you know, it's a humbling thought. We are redundant, but there's a lot of stuff that we could have a huge impact if we could sort it out. And so when you're thinking about your science, first, please share your data. Second, think about this. I mean, it's hard because the system doesn't help. You know, as much as you want to work on this, the grant system won't let you. As much as you want to publish a paper down here, the peer reviewers will nail you. It's not important, right? So it is hard. I have to play this system. You have to play this system. But if you can figure out a way, if you get a list of proteins from your microarray that look cool, choose the one that nobody works on, right? Don't choose the one that everyone works on. Because, oh, I know something about that. I'm going to work on that one because everyone's going to work on it.